سعدني ان ارحب بحضراتكم في ثالث للمؤتمر الجمعيه المصريه الامريكيه. Our first speaker دكتور سامح علي جينا من محاضره entitled Metabolic Reprogramming and Metabolic Strategy for Cancer Treatment. تفضل.
صباح الخير. I'd like to start by apologizing for coming late. As you know, Metabolic is always having some issues. I just should apologize first. It's an honor to be here, and I should thank all the organizing committee for inviting me to come to share with you some of an exciting new field, relatively new field. There's no new fields in science, as all of you know, but it's just a field that's really emerging quite recently on how to manipulate cancer survival through manipulation of cancer cells or manipulation of metabolic activities in cancer cells. I should probably start by warming up by just giving you an idea of what kind of sequence I'm going to be talking about cancer first, and then talk about the hallmarks of cancer progression, and this is going to pave the way of understanding why we're targeting metabolism and what kind of metabolic pathways are important in cancer. The next section, I'm going to be talking about how to target metabolism and what kind of instrumental approach or what kind of experimental techniques are being used in Zoology City, in the center that I'm directing. And I'm going to share with you some of our recent results. It's not going to be extensive because these results are under development. We are trying to collect more results and hopefully be able to put together our first experimental publication from Zoology City soon, inshallah. I'm going to close my challenge and my future directions, but I don't know exactly. Probably this is just like a soccer or football player who's warming up. Warming up is good for him, but not for the spectacles. So cancer is essentially a large group of diseases characterized by uncontrolled growth and spread of abnormal cells. Of course, these are just the expressions or the definitions that we're going to talk about for the rest of this talk. Neoplasm is a new growth of tissue that serves no physiological function. Tumor is a clumping of new plastic cells. Malignant is cancerous, and benign is non-cancerous. Metastasis is malignant tumors that are not enclosed in a protective capsule, have the ability to spread to other organs, and mutant cells are those cells that are including disruption of RNA and DNA within normal cells, and they may produce cells that differ in form, quality, and function from normal cells. Cancer is, as you all know, is an extremely big problem in the whole world, and of course, there's no statistics that are there's no accurate statistics about Egypt. So usually, when anyone is talking about anything, we start by talking about the statistics in the United States. So approximately about 700,000 persons, they die every year by different cancer types. And the relatively good news is that recently, starting from mid-90s, there is some sort of stability or even reduction in the rate of death due to cancer. And in fact, in men, cancer is more prevalent. And for example, during the period from 2008 to 2010, almost 1 in every 2 men were diagnosed for a specific or certain type of cancer. This is from American Cancer Society. In Egypt, and this is one of the main causes that allowed me, or that actually invited me to work on cancer, even though I'm actually originally much more focusing on aging and on nervous system aging specifically. But in Egypt, cancer is an extremely important problem. It is very clear. Sorry. it's very clear in Egypt that cancers, including liver cancer, especially because it's directly implied upon hepatitis C infections. Also, we have issues in lung cancer and all breast cancers. These are putting us among the first nations in the rate of cancer incidence. So what causes cancer? The cancer is caused by either external factors such as chemicals, viruses, radiations, and lifestyle, and internal factors such as hormonal imbalance, immune conditions, and inherited mutations. There are a couple of theories about the origin of cancer. I don't think that there is time to talk about these in details, but we will see that in cancer cells, something happens to the cell cycle. So cell cycle is having some checkpoints. These checkpoints make sure and give signals for the cells to continue and to divide or to replicate its DNA. But in cancer cells, these checkpoints are actually lost. And this causes, instead of just a normal cell division, it actually causes some sort of division that's uncontrolled, leading to tumor and eventually cancer. In another word, also just relative to normal growth control, where if you have some sort of mutation that happens, for example, upon cell division, what happens is that these cells are usually undergo programmed cell death. But in cancer cells, these cells keep dividing, and as they divide cells or the daughter cells will probably acquire more mutations. There are thousands of mutations that are associated with every type of cancer. And that's because of the evasion of these cancer cells, and they are not undergoing apoptosis. So, if we're going to talk about specifics of cancer, what happens, especially in relevance to what I'm going to discuss with you about our results, is first, metabolism is going to be altered. There are some definitely limited supply of oxygen because of tumor progression. There is also an avoidance of using mitochondria as a source of energy in cancer in tumors. And also there is an enhanced glucose uptake. These are the three different hallmarks of altered metabolism in cancer. At the same time, there is also oxidative stress. And oxidative stress is actually all over the place. If you go through the literature, people are usually reporting that in cancer cells there is elevated markers of oxidative stress. Some others are actually reported reduced levels of oxidative stress. These are things that are under investigation. We are also required to study these. And the initial conclusion here, we still don't have sufficient data to talk about, but we actually think that intracellular reduction in oxidative stress happens in cancer cells, while extracellular loss release is essentially very important in recruiting or actually restructuring of the stroma or the cells around tumors. Finally, these reactive oxygen species, they cause DNA damage. DNA damage, as you know, can cause uncontrolled proliferation and metastasis. Okay. So here in this slide, I think it's a little bit, I don't know whether you can see it from back there, but it's very also intelligent kind of slide. This slide describes the processes of metabolism in cells, generally speaking, and there are some pathways that are much more relevant to cancer. For instance, if we look in cancer cells, you will see that glucose transporters are overexpressed. And these are some of the markers of tumors. And the pathway, the normal pathway of cancer, involves multi-step metabolic transformations of calcium, of glucose, leading eventually to pyruvate. This is a very important intermediate in the metabolic pathways in cancer cells and in normal cells as well. In normal cells, pyruvate usually, or it's much more likely that pyruvate would enter mitochondria and then would join the DCA cycle. Whereas in cancer cells, it is usually converted into lactate, so in what's so-called glycolytic pathways. In cancer cells, as we will see, there is an alternative way also for supporting metabolic demands, and these are through, this is the DCA cycle inside mitochondria, and there is also the other way of metabolism is the glutamine metabolism. Glutamine metabolism is an extremely or an increasingly important metabolic pathway that's now implicated in cancer, and it is very well known now that inhibiting glutamine metabolism would have a very important role on regression of tumors. And this is because glutamine metabolism essentially is used to synthesize lipids, and also in addition to other pathways, these provide ways to synthesize nucleotides, proteins, and lipids that are extremely important for proliferating cells. Okay, so in metabolic pathways in cancer, in normal cells, usually what happens is that glucose is entering cells and then enters the TCA cycle through pyruvate, and then this mostly is converted into ATP, which is important for supporting cellular processes. Whereas in cancer cells, what happens is that glucose is essentially converted into biomass. And this biomass transformation happens through the pathways that I've been talking with you about. The biomass is important for tumor progression, for spreading of cancer cells, for the synthesis of components of the cells, including cytoskeleton, including membranes, and so on. So essentially, also for just to distinguish between normal cells and proliferating tissues or tumors, what happens is that most of the energy demand is obtained in normal cells through pyruvate metabolism, either to go into mitochondria and produce ATP, or small amounts, relatively small amounts, are actually converted into lactate in what's so-called anaerobic glycolysis. For anaerobic glycolysis, 2 mole of ATP are produced per 1 mole of glucose. For oxidative phosphorylation happening in mitochondria, almost 56 moles ATP are produced with each mole of glucose, indicating how efficient mitochondria is. If you have a proliferating tissue or a tumor, what happens is that you will have a switch into lactate production, a switch into what's called aerobic glycolysis or the Warburg effect. In this, 4 moles of ATP are produced per 1 mole of glucose. Okay, so in normal cells, if you surprise yourself or surprise your muscles, for instance, with a sudden demand like going and doing exercise or running after a bus or something like this, what happens is that most of the energy needs starts by utilizing glycolytic pathways because it is extremely fast, it produces large amount of energy immediately. But then you cannot afford doing this for a long time because glycolysis produces lactic acid, and lactic acid may just acidosis, as you know, and this can cause some damage to cells. So what happens is that mitochondria kicks in gradually, and then they take over as the main source of energy. In cancer cells, what happens is that both glycolysis and oxidative phosphorylation are continuously contributing to the energy demands of cancer cells. So the hypothesis of this work, or the metabolic interventions, essentially is based on what would happen, the question of what would happen if we apply metabolic switching. What would happen if we essentially convert this kind of form of metabolism back into the normal mode of metabolism? This, of course, could be, if successful, it could be of great implications for cancer. Why? Because essentially you don't need to design specific delivery or anything like this. Because if you, for example, you switch back cancer cells into normal mode, now you're enhancing the normal metabolism in normal cells. So there is expected no or probably minimized side effects for these kind of interventions. Okay. So it is important just to outline why this hypothesis has been made. It is very well known now in literature that oncogenic signaling and tumor cell proliferation or metabolism are very closely interrelated. They are usually, there are some very good connection or very significant connection between oncogenic signaling and between metabolism. Also, the malignant transformations, they are essentially, they are going through a bottleneck in which metabolism is actually changing. So there are some really important and fundamental changes in metabolic pathways. Other, if it doesn't happen, if it doesn't happen, there is no way that a cancer cell is formed. So it indicates how important metabolism or in cancer transformations. The other thing, or the other observation in literature is that all genes involved, almost all genes involved in mitochondria or mitochondrial metabolism, they are known to also exhibit a sort of tumor suppression function. So they are all tumor suppressors, indicating that there is a big choice for cancer cells to survive. You have to silence mitochondria because most of the mitochondrial genes are tumor suppressors. Okay, so again, another thing that's really very important is that mitochondria are really important for a checkpoint for apoptosis. So it makes a lot of sense to try to eliminate mitochondria from the equation, just trying to avoid any apoptotic response. So the conclusion here is that cancer cell survival involves the minimization of mitochondrial function. Now, what we are trying to do is to try to switch this back. Let's try to activate mitochondria in cancer cells. If you activate mitochondria in normal cells, that's well and good. This is something that we should probably be happy about. But if you do this in cancer cells, what you would expect is that you will see that cancer cells are unstable. Okay, so the hypothesis is that in order for us to revert tumor progression, we probably are able to design some sort of metabolic manipulations to prevent tumor progression. Before delving into some of our data, I'd like to just share with you what are the ways and methods that we use to evaluate mitochondrial function, especially in tissues, because we are also planning to work with human tissues. The other thing also is how to evaluate mitochondrial pathways such as oxidative phosphorylation by mitochondria or glycolysis or glutamine metabolism. How do we do this? How do we actually evaluate this? So a couple of slides are telling you something about what happens or about what we do to evaluate mitochondrial function. In the center that I've established in Twin City, we have multiple cores. We have a core for metabolism. I will show you more about this core. We have also imaging and spectroscopy and biophysics core. We have cellular and molecular biology lab, and we are now under construction of an electrophysiology lab as well as behavioral study lab. We have also an animal facility that we use for generating cancer models. We do have all of these equipments installed and running, and they are covering a wide range of biological subjects, starting from molecules such as reactive oxygen species and up to studying a whole organism. Okay. So I think I'm going to skip through this because probably I was late, but the most important take-home message is that mitochondria is important. And that's why this is the past decade was actually declared as a decade of mitochondria, as the role of mitochondria in so many diseases, including neurodegenerative diseases, obesity, and diabetes, aging, and of course, importantly, in cancer. That's why we established a state-of-the-art cutting-edge facility in Twin City to study mitochondria. And in order for anyone to study mitochondria, you need to evaluate different aspects of mitochondrial function. First thing is to study respiration, how mitochondria is utilizing oxygen, because aerobic metabolism involves the consumption of oxygen. We are equipped with tools that will allow us to study oxygen consumption at the level of a picomole, which is really something that's extremely sensitive. The other thing that that you should work on is also not only just consumption of oxygen, but how this oxygen is converted, whether this oxygen is converted into ATP, or is it converted into reactive oxygen species, or anything else. And this is also something that we study in great detail in the center. The other thing that we probably require to study also is the reactive oxygen species. Reactive oxygen species, as we know, are very important not only in cancer but also in aging, in neurodegenerative diseases. And we actually are equipped with the only system, the only technique that is capable of detecting and evaluating and identifying free radicals and studying the dynamics in cells and in isolated mitochondria and so on. The next thing to study is how mitochondria is contributing to signaling pathways. And of course, relevant to cancer biology, of course, the apoptosis or extreme apoptotic signaling is extremely important subject to study. And this is one of the things that we do regularly. Of course, also mitochondria is important. The structure of mitochondria morphology is important. The dynamics are important. Where mitochondria is continuously under fission and fusion, we also try to study these in different cell models. In the laboratory for metabolic studies, we have all of these components. These are the three most important components. The first one here, this system is called the seahorse, and the seahorse system is a plate reader that is equipped to measure both oxygen consumption and also the lactic acid release in a very, very high precision. And this would allow us to study both mitochondrial function or oxidative phosphorylation and also lactic acid or glycolytic pathways in cancer cells while infusing drugs. So we can actually, essentially online, we can infuse drugs dramatically and we can see what does these drugs do, as I will show you some of the details. The other system here is an electropolymagnetic resonance spectrometer. This is, I was telling you about, this is a system that detects free radicals. Detects free radicals also can be used to study membrane dynamics, how membranes are rigid, how they are changing in disease models or aging, and so on. The last but not least is the Ouroboros system, which because of the power of this system, we are actually thinking of buying another copy of the system because this is essentially the only system that enable you to study in great details mitochondrial function in intact tissue. So you essentially can get biopsies from humans, from animals, and you can study mitochondrial function, but at the same time, as I will explain later, you can also detect a lot of other components to study or to complement your studies. This is the Ouroboros system that I'm telling you about. It has electrochemical polarigraphic sensors to detect oxygen and also to detect changes down to million pH units that will enable you to understand how lactic acid is released in cancer cells and others. The other thing is also that this system is capable of measuring, as I was explaining, mitochondrial function even in biopsy. And it's a pretty straightforward procedure. You can prepare fiber bundles and then you can place them and then you can study what's so-called multiple substrate protocol. And this protocol will allow you not only to study the general mitochondrial respiration, but also each individual complex on electrotransport chain complexes. It would allow you to study each of these complexes' function. And this is quite important for several diseases. For instance, we started to understand that complex 2 in mitochondria is very important to cancer cells. And that's something that we are using this system to study. I can just give you an example on how to use this system. In fact, you can evaluate mitochondrial function. Here, comparing a normal heart and diabetic heart, you can see that the blue line indicates changes in oxygen levels, and the red line is indicating the rate of change of oxygen consumption. As you can see, the higher the level, it means that more efficient mitochondria is, and you can see that normal heart mitochondria is much more efficient than diabetic heart. The other thing that this system can also detect is what's called the maximal respiratory capacity. This is important also to cancer because you know that, for example, your muscles are having a maximum respiratory capacity, maximum efficiency. It's not used, but it is many disease states are characterized by lower maximum respiratory capacity. Means that you cannot really respond to stress. Your system cannot really respond to stress. So this parameter is an important parameter, physiologic parameter that we can detect it. For instance, the maximum respiratory capacity in a normal heart is higher than diabetic heart, and we confirm that this is statistically significant. Okay. The other, I'm sorry about the that part of the slide, but you can also, while monitoring the respiratory function, you can also monitor hydrogen peroxide release, the reactive oxygen species release from mitochondria. And this can happen through a system that can be combined with that system. It's a fluorescence detection. So you can use fluorescence detection in the same sample while adding substrates. And we have been able to show how mitochondria and cancer cells are producing hydrogen peroxide. This is the photograph of the module, the fluorescence module that you can use. This is a list of all things that you can detect. Hydrogen peroxide, you can detect mitochondrial membrane potential, you can detect ATP, you can detect calcium in your samples. This enables us to essentially study a lot of things about mitochondria. Metabolism in general is extremely complex and intricate process. It involves a lot of things. A lot of pathways. So the seahorse system is capable of measuring both oxygen consumption rate, and it's also capable of measuring extracellular acidification rate. And this will enable us to study a lot of subjects. I'd like to just focus on cancer because this is the main take-home message of this talk, that what we need to do, essentially, what we thought about when we started this kind of research, is we need to target mitochondria, we need to target glycolysis, and we need to target glutamine metabolism. And essentially what we did is we, based on some literature hints, we wrote a library of chemical compounds. And these chemical compounds are original natural products that are characterized with the Egyptian environment as well. Just hoping that if we could find a target, we can probably have something that's actually novel. And we screened all of these drugs based on their effects on cancer cells. First by cytotoxicity assays, which is quite common, and then second by their effects or their interventions or their interaction with metabolism. And then we selected certain compounds to continue our study with. So the essential requirements for a metabolic interventional pathway that will probably kill cancer cell is to inhibit glycolytic pathways, because as I was explaining, in cancer cells, glycolytic pathways are much more active, while leaving mitochondria alone, or even activating mitochondria. Normally, when you inhibit glycolysis, there is an automatic activation of mitochondria because of course, of energy needs. So these are list of chemical compounds that we tested, of course, in addition to many other compounds that are obtained from natural products in Egypt. And each of them are targeting specific enzyme in the pathway, metabolic pathways of glycolysis. And we focused on iodoacetate just as a test compound, as a control compound. And this is what I'm going to show you today. Again, also mitochondria, we understand in great detail how to inhibit mitochondria and how to manipulate mitochondrial metabolism. We know exactly inhibitions of each complex can be carried out just to try to adjust your conditions and try to find the exact compound and the function of that compound. So the first step to understand how to tackle metabolism in a given type of cancer is essentially you use cells in vitro, you bring some other type of cancer cells, and you monitor the ratio of oxygen consumption rate relative to an extracellular protein release or acidification rate. And then by phenotyping these kind of cells, for example, if you find that the race or the relationship lies in this range, this indicates that there is much more glycolytic activity in those cells. Then it is important to find molecules that can inhibit glycolytic pathways. However, if you see that they have more OCR and less ECAR, this means that you should probably start by targeting mitochondria itself. If you have it much higher, this means that your cells are relying much more on fatty acid oxidation. And this is quite important in cancer cells as well. When you are proliferating, you need a lot of energy, and in this case also pulmonary oxidation is used to understand whether fatty acid oxidation is important or not. Finally, if you have a very high ECAR and also OCR, this indicates what's so-called a state of uncoupling. Your mitochondria is really uncoupled. So there are chemical compounds that dissipate the energy that's produced by mitochondria. So what you do essentially is you try to first screen your cells to identify the phenotype. Okay, and this is an example of how to do this. You just repeat the acquisition of ECAR and versus OCR, and then you determine whether these, for example, these cells are much more active in mitochondria, whereas some of other cells, like for example, this cell, for instance, is high on ECAR, okay, or this cell is high on ECAR, and then after determining these, you start to outline your strategy. What kind of metabolic pathways you will be targeting. So we started by working on liver cancer for, of course, obvious reasons in Egypt, and we essentially tried to screen some compounds, and the compounds, there are some criteria we fulfilled, and iodoacetate actually fulfilled this criteria. So if you see, this is the percentage change in oxygen consumption rate, and this is the percentage change in the extracellular acidification rate, the low one contracted. Okay? So essentially, by using this compound, we could inhibit almost completely, we could inhibit the glycolysis, while essentially slightly enhancing mitochondrial function as well. And of course, we screened many other compounds. I'm just not going to go in details. And this is another compound that's actually not iodoacetate, but we are still working on this compound, and not allowed actually to state the structure of that compound. But this compound is used for cytotoxicity and also for inhibition, and we could find that this compound can inhibit glycolysis while enhancing mitochondrial function. So now we know that there are some compounds that fulfill the criteria that we put to ourselves. So now let's try them on cancer cells. Again, these are just telling us that applying these compounds, for instance, here, you can see that when we treat with drug A or B or C, we could see up to 80 or more than 80% of cell death. So in liver cancer, if you use this strategy, you essentially can kill more than 80%. And the nice thing about this is that we are working with relatively low concentrations. The treatment time also is less than 4 hours. You don't have to incubate for days as it's usually done. And what we are doing is we are incubating for 4 hours, and then we are monitoring cell death and cell viability. We are monitoring apoptotic pathways, and it's actually starting apoptosis very early on. And this is because, of course, you are depriving cells from their energy needs while activating mitochondria. Why is it important to activate mitochondria? Essentially, it is important, in my opinion, because when mitochondria are shielded in cancer cells, they are not really producing that much energy, or they are actually devoting energy to hexokinase. Hexokinase is an enzyme that is used to convert glucose into glucose 6-phosphate. And this essentially charges glucose and keeps it inside the cell, and also it keeps the gradient of glucose to happen all the time. So the glucose uptake is increased to fulfill the needs for glycolysis. But when you activate mitochondria, essentially these mitochondria are not really ready to produce energy. So what they do is actually they produce active oxygen species. And we confirmed this by flow cytometry, as you can see here, that by flow cytometry, if you treat with iodoacetate and other compounds that we use, you have an enhancement. and the intensity, and also a state for more cells with reactive oxygen species. Reactive oxygen species, they trigger apoptosis. You also put, I'm sorry, this is off. You cannot see anything. But essentially what I'm trying to show you is that there is also a quiescence detection of reactive oxygen species that increases upon this kind of treatment. What happens to these cells is essentially also they undergo growth arrest. And you can see the concentration dependence of iodoacetate, even starting from 10 micromolar. You can see a complete stop, a complete halt of cell growth. And after 4 hours, you can see that cells are really different at their time. Essentially, this would be associated also with apoptosis markers early on. Starting even from about 4 to 6 hours, you can see apoptotic markers. Okay, so I'm gonna just close here by saying that I just couldn't get the chance to show all what we have done, but this is just a short story just to tell you that this kind of strategy was successful in killing cancer cells in vitro. And it was very efficient, and also we have some evidence and indications that the acts or the adverse effects on other types of cells are actually applying the same treatment on fibroblasts as a control system, just to see whether they would have some adverse effects on normal cells or not. And what we're seeing is that, of course, there are some induction of cell death, but it cannot be compared with what happens in cancer cells. So this is the first thing. The next step that we are actually working on is the discovery of certain new and novel drugs, and then applying this into animal models now, so just to try to see whether these treatments are going to reverse tumor progression or not. At the same time, I'm also in the process of negotiation with colleagues with clinical samples. We would like to analyze the tumors for metabolic pathways, just to get some clues whether what kind of just a strategy. You remember that I was telling you I'd like to know exactly the relative relationship between extracellular acidification rate and also oxygen consumption rate. I'd like to know also in human tumors, and if this is different in one tumor to another one or not. Just to try to be able to optimize and select the actual and the best strategy to go. I'd like to thank my group, of course, and of course Dr. Ahmed Zuhair and Dr. Sharif, who gave me the opportunity to come back from the University of California, San Diego, and to establish this very, very challenging kind of project. But it's actually very exciting. And of course, with challenges comes always excitement, and comes a lot of things like the reward itself is very high if we are successful. So I'm also very willing to answer any question and also to discuss any future collaboration with any one of you. And thank you so much for your kind attention.
Thanks, Dr. Samah. Any question, please?
Please, I want to ask who about the compounds used as antioxidant is synthetic or natural?
We didn't use any compound. We didn't use any compound as an antioxidant. We used compounds to intervene with this compound here. You probably meant this. I was trying to say that using this treatment that activates mitochondria produces more free radicals. So in cancer cells, you actually want to produce more free radicals, not to eliminate free radicals. Because free radicals, they are very early signaling molecules that induces apoptosis. So I'd like to enhance free radicals in cancer cells, not to prevent them. That's why I was always kind of confused, because many people are trying to use antioxidants to treat cancer, which is my opinion.
But when you activate the free radicals productions, maybe exert adverse effects on another normal cells?
Right, that's exactly what we're trying to understand now. What the characteristics of the active oxygen species is that when they are produced inside the cell, it is unlikely that they will leave the cell because they are highly reactive. They only leave the cell if they are converted into hydrogen peroxide. And of course, hydrogen peroxide is a signaling molecule as well. So what we are trying to do is actually control the amount of free radicals within cancer cells and And as I'm saying, is that this happens in normal cells, and actually in cancer cells, activating mitochondria produce more free radicals. But activating mitochondria in normal cells, they don't produce that much free radicals. Because there is a normal mode of metabolism for mitochondria. Mitochondria is usually producing ATP while minimizing the amount of free radicals that are produced in normal cells. But in cancer cells, they are undergoing certain problems, and if you activate them, they produce more free radicals.
حضرتك بتمشي على المحاضرة الكبيرة المعلومات الحلوة. حضرتك تقول عشان تشطب كانسر وماس اكتيفيت ميتوكوندريا. Like more breathing oxygen?
Well, you know, I missed some of the slides I was preparing. That actually about 30% of suspected causes of cancer is due to lifestyle. And now it's very well established that the lifestyle, including dietary, including activity pattern, is essentially very critical for the high incidence of cancer. In fact, we are going into a specific project with the European community in which we are testing a hypothesis of combining specific interventions for metabolism with exercise to try to combat cancer. And the idea is that if you are more active, you probably would essentially resist tumor progression. And so that probably it has something to do with mitochondrial activation as well.
سمعنا ان في ناس بتاخد الاكسجين يرجعوه وحاجات كده والاوزون. هل في طرق حاجات بتعمل؟
والحقيقه يعني في حاجات كتير قوي بتتقال في الميديا والحاجات دي ما انزل الله بيها سلطان. يعني الحقيقه انا اقول لحضرتك ان هو الفري راديكالز مطلوب على الراديكالز. It's actually much easier to form بس the target we cannot go that far. يعني الاوزون مثلا الاوزون is actually producing single oxygen. هو الناس بتقول يعني ان هو بيستخدم في حالات معينه بتاعت مثلا في ناس بتقول لك ان انت مثلا ممكن تكوني تستخدمي بيور اوكسجين تعملي اللنج بتاعتك الهايبر اوكسجين. الحقيقه الاكسبوجر للهايبر اوكسجين برضه از نوت ريسك فري لان الاكسجين is toxic اصلا. الاكسجين اصلا is toxic because it produces more free radicals. And essentially, if you expose yourself to too much oxygen or ozone, you probably would have adverse effects on normal tissue, would have oxidative stress state, and probably at certain stages it can actually induce cancer. So that's why things has to be done in a scientific way rather than just trying things for a change.
I would like to ask you, thank you very much for such a very interesting وعليكم السلام.
يعني هو الشرف ليا ان انا اكون موجود معاكم.
والله هو الشرف بتاعنا احنا يا فندم. يعني I heard a lot about you and it's a real pleasure to meet with you at last. So I hope this is the beginning of a long cooperation with our faculty and with our society, inshallah. The question that I would like to ask is during your presentation, you said something about that you have an animal facility. And I also saw in your slides that you are doing most of your work on animal models. I would like to ask, is your animal facility according to the international guidelines?
No.
Do you have, okay, let me continue this and then we can cooperate later on. Do you have an ICOC?
We are in the process of establishing an ICOC committee. However, yes, of course, this is an extremely important question. And I'm not willing to publish anything before we establish an ICOC committee or to really establish a good, high-quality standard animal facility. As you know, as you all know, we have been going through a lot of difficulties in Zoology, as you know. And we have to leave our buildings now and we have to go for the new one. In fact, I have a meeting just after this meeting with the engineers. We're talking about the design of what would be, inshallah, the biggest animal facility in the Middle East. We are in devoting a whole basement floor for a whole building to establish an animal facility. And this is going to be, of course, going to be one of the biggest animal facilities in the region. But for the time being, and for the purpose of carrying out experiments and also training my students, I modified the space. I applied as much as I can. إن شاء الله حضرتك تشرفينا وتشوفي اللي أنا عملته يعني. I tried as much as I can to make a clean area for animals and for animal treatments. And also I divided and I also control the humidity, I control the temperature, I control the light cycle. All of these things are established in the small animal facility that I established right now as a transient animal facility. I cannot claim that this is up to the standard, international standard, and that's why I'm not willing to publish anything before we actually establish a real animal facility. And of course, I'll be really very happy if you can help us with this.
You shouldn't not only publish, you shouldn't even start your experiments, animal experiments, except when you have an ICOP and an animal facility that the ICOP can approve of. Because I believe this is one of the most challenging issues that we have now in Egypt and the Middle East. And I believe Mr. Bas Straub is going to be talking to us about international publishing.
Right.
So yes, I would like to cooperate with you in this regard. In Cairo University, the Faculty of Science, Cairo University, 3 years ago, we have established the first ICOC in the Middle East. And we had a lot of opposition. I believe that now they have started to realize the importance of the ICOC. Because without it, it's not, you cannot publish, you cannot start your experiment and then go and have your approval. This is unethical. And this is what we have stopped doing. Starting in September, we said no. We're not going to do this because this is unethical. And we have to keep our credibility, not only in our faculty and our university, but we have a problem in that we have to keep the credibility from Egypt as a country, because now we are facing humongous challenges in international publishing regarding this. So I would have thought, since you came from San Diego, and they there apply the guidelines rigorously.
I am a member in the ICOP committee in the UCSD School of Medicine.
Is that so?
Yes.
So could I use you also in our ICOP?
Absolutely. Absolutely. And of course, I thank you so much for your comments, and we agree, and fully agree with you and with your comments. And definitely I'm willing to collaborate and cooperate in this regard. And definitely animal handling and animal treatment is an extraordinarily important issue that we have to establish as a culture in Egypt. And this is definitely one of my goals when I returned from the University of California. Of course, I gave my students all training that I have went through when I started my biomedical research. And also, I'm in the process of inviting the head of the ICOP committee in UCSD, the University of California, San Diego, to help me establishing the essential procedure for approval, and protection. And I will be, of course, very happy to collaborate with you. Thank you so much.
Because we are also with a group from the Cairo University, around a group of 7 or 8. No more. We went to Germany. We were 27 to director of Aachen University, and you know that the Aachen director of the Aachen Institute for Animal Science, Experimental Surgery and Animal Science is the co-director or something like that, or deputy vice chairman of the KILASA, which is something like the ALAC in the United States, not ALAC very much, like let's say the National Academy of Sciences in Europe. And we also went to the United States 3 times to the biggest ACOG conference there that is undertaken by the Primandar. So we did have rigorous training, and we have also applied in Cairo University this year that no thesis or any research will be undertaken or will even start except when the researchers would go through a training in the, so we do have some, so I would like very much to cooperate with you in this regard, because we need to spread this kind of awareness to all our colleagues in the different universities.
Sure, and I'm
so
also willing to have.
Thank you very much, Dr. Selma. Thank you.